to prevent the self-pollination, right? Why there is a need to prevent self-pollination, guys? It is because, when the self-pollination takes place, self-pollination means like the same flower having both male and female gametes, right? And what happened when the same flower have both male and female, then the pollen grain coming out from same anther and reaches to the stigma of same flower. So again the same progeny will coming in the next generation. So no genetic variation you will see in the next generation. Like in the sexual reproduction, you will see the genetic variation only, na? But here no genetic variation in the self-fertilization. It will becomes like asexual reproduction only. In case of cross-fertilization, there you will see what? There you will see your variation. How? Because suppose pollen grain coming out from one species reaches to the flower of other species or the same species of different kind. So there you will get a variability, genetic variability, which is good for evolution, right? So to prevent the self-fertilization, the outbreeding devices we are using. Now, what are the outbreeding devices? What are the methods to prevent the self-fertilization? Pollen releases in stigma, receptivity not synchronized. So what does it mean? That suppose what happened? Pollen releases, but stigma is not mature till then, right? So this is one of the method कि okay, pollen तो आ गया लेकिन अभी stigma mature ही नहीं है, तो that will prevent the what? Self-fertilization. भाई दोनों mature होंगे, then only fertilization takes place ना. So pollen releases, but at the same flower stigma not mature. So this is one of the method. Anther and stigma are placed at different position. So in a same flower, we just place the anther and stigma at a different position so that they are not, the pollen grain are not able to reach to the stigma. Inhibiting pollen germinating in pistil. Now again the germination of pollen also get inhibited. Production of unisexual flowers. So now bisexual and unisexual flower, what is the difference between bi and uni? Uni means only a flower having only a single sex, bisexual having both the anther and filament, both the stamen and the pistil. So now inhibiting, or you can say the production of unisexual flower. If the flower is bisexual, then there is a chance to undergo the self-fertilization. Right. So in that case, what happened? Flower should not be bisexual, it should be unisexual. So a unisexual flower encourages cross-fertilization. Clear? So these are some of the outbreeding methods. Then we have a pollen pistil interaction. What do you mean by pollen pistil interaction, guys? The pistil has ability to recognize the compatible pollen to initiate post-pollination event that leads to fertilization. Now suppose what happened? Pollen from a flower, okay, reaches to the stigma of another flower. Now the stigma has capability to recognize whether the pollen is coming from the same species or pollen coming out from the another species of a flower. Okay, like suppose a pollen reaches to the rose flower. So the rose flower stigma has a capacity to identify that whether it is coming from rose plant or it is coming from a sunflower. So this is the capability of the stigma is having. Okay, if it is of same species, then only the stigma allows the pollen grain to stick over their surface and to germinate the pollen tube, and the further procedure takes place. If it is not of same species, stigma, what does the stigma will do? Stigma reject. Stigma says, no, this pollen grain is not from my species. Okay, so I'm not going to get fertilized with this pollen. So pollen grain produce pollen tube through germ pore to facilitate transfer of male gamete to embryo sac. Now artificial hybridization. Artificial hybridization means what, guys? What do you mean by artificial hybridization? Crossing different varieties. Sorry, crossing different varieties of species, hybrid individual with desirable character of the parent plant, desired pollen grain for pollination, stigma protected from contamination. So basically what happened, artificial hybridization means now you are crossing different species of pollen to different species of stigma, right? Or you can say that artificial hybridization is basically the mean of cross fertilization. ओके आप उनको क्रॉस फर्टिलाइजेशन करने के लिए मजबूर कर रहे हैं. दिस इज व्हाट आर्टिफिशियल hybridization. Crossing different variety of species, creating hybrid individual with a desirable character, like whatever the character you want of the parent plant, desired pollen grain for pollination, stigma protected from the contamination. Emasculation, removal of anther. So what happened, how you are going to perform this activity, guys? What you need to do, you need to just cut the anther. So like suppose you have a unisexual flower and in that flower you have a stamen and you have a pistil both. So when you cut the anther, that process, cutting the anther process is called as emasculation. ओके आपने एंथर को काट दिया, अभी एंथर है ही नहीं, नो पॉलन ग्रेन फॉर्म, नो फर्टिलाइजेशन टेक्स प्लेस, ओके, नो पॉलिनेशन टेक्स प्लेस. सो दिस प्रोसेस इज कॉल्ड emasculation. So it's not like you just left the flower after cutting the anther. Okay, so now you have to protect the stigma so that no other flower pollen grain also reach to the surface of a stigma. You have to protect also protect the stigma. So what you did, you just cover the flower with a paper, process called as bagging. Okay, now what you did, you just stick the pollen grain of desired character. Like अब आपको जो भी character चाहिए plant में, right? You just put the pollen grain of that desired character of the stigma, cover the flower so that no other pollen grain can contaminate it, and then you will get the desired plant. So this is what we call artificial hybridization. Clear? Artificial hybridization understood, everyone? Now the most important is double fertilization. Guys, double fertilization is something from where you will get questions directly in your exam every time. Okay. The double fertilization is the it is a characteristic of angiospermic plant. Okay, double fertilization is the characteristic of angiospermic plant. Okay, gymnosperm do not undergo double fertilization, no other plant, only angiospermic flower can undergo double fertilization. Now why it is called double fertilization, what happens in double fertilization, let us understand. See, this is what, this is your female gametophyte. This is your female gametophyte, or you can say female reproductive part called as pistil. Okay, now you know that this pistil is having three parts. What are those? This is your stigma, right? This tube, style, ovary. Okay, so what happened? Now this is the pollen grain, right? The pollen grain is having two sperm nuclei. The pollen grain is having what? two sperm nuclei, okay, and a pollen tube nucleus. So this is the germ pore which actually converted into a pollen tube, and this is the pollen sporolanin wall which protect the pollen grain, right? So now what happened? This pollen grain reaches to the stigma, okay, pollen grain क्या किया? Stigma पे आके stick हो गया. Now the pollen grain started forming tube, pollen tube. So what is this actually? This is a style. This is what? Pollen tube. So now the pollen grain forms a pollen tube. This pollen tube, through style, it enters into the ovary and reaches to the micropylar end of ovule. Okay. Now this is your ovule, female gametophyte, or you can say ovule. Okay. So now within the ovule, what you have? You have 3 antipodal cells, 2 polar nuclei, and 1 egg cell and 2 synergid cells, right? And this opening is what? Micropylar opening. So the pollen tube reaches to the micropylar end. See, this is the pollen tube. Pollen tube is having sperm nuclei. How many? 2 sperm nuclei, okay? And here inside the egg, you have 1 egg nuclei, 2 synergids, 3 antipodals, and 2 polar nuclei, right? 6, total 8 nuclei you have within them. Clear? Now this pollen tube reaches towards the micropylar end. This end is what? Micropylar end. So through the micropylar end, the pollen tube enters within the ovule. Now what happened? Since it has 2 sperm nuclei, so 1 sperm nuclei fuses with egg and the other sperm nuclei fuses with both what? Central nuclei. Okay. Further what happened? Now the egg fuses with sperm nuclei, forms zygote, right? Diploid zygote, and the another sperm nuclei which fuses with the 2 central nuclei, central cells, right? What it causes? What it forms? It forms endosperm. ठीक है? It forms endosperm. Am I clear to everyone? Here diploid zygote forms and a triploid cell forms. Now ultimately this diploid zygote converted into embryo and this triploid cell forms endosperm, right? And ultimately you will get a complete seed. So now here, since fertilization takes place 2 times, first the sperm nuclei fuses with egg, secondly the sperm nuclei fuses with polar nuclei, it is called as a double fertilization. Am I clear? Now see what happened here. At the very first sperm nuclei, which is haploid, okay, which is haploid. Haploid means only single set of chromosome, 23 number of chromosome, okay. First sperm nuclei fuses with 1 egg nuclei. Now egg nuclei is also what? Gamete, and both the gametes are always haploid. Whether it is male or female gamete, egg or sperm nuclei, always they are haploid. So when they both fuses together, process called syngamy, okay. And then what happened? What you will get ultimately? You will get zygote, and this zygote is what? Diploid, which is 2n. This zygote is what? Diploid in nature. Why it is diploid? 1 n from sperm, another n from egg. You will get a zygote which is diploid, okay. Now the second sperm nuclei fuses with, again the second sperm nuclei is also haploid, fuses with two polar nuclei, right? Now two polar nuclei, N and N, okay, two polar nuclei, N and N. What ultimately you will get? See here, 3 nuclei fuses here, right? 3 nuclei fuses here, process called as triple fusion. 3 nuclei fuses, process called as a triple fusion. And ultimately what you will form here? Ultimately you will get endosperm. Now how many nuclei this endosperm is having? 3 nuclei, right? So it is 3N, or what you can say, triploid, right? So they are what? Haploid. You will get endosperm, diploid. Further, this zygote converted into embryo, okay? This endosperm provide nutrition to this embryo, and ultimately what you will, this embryo converted into seed, right? Ultimately what you will get? You will get seed. So this is the process. Since 2 times fertilization takes place, first syngamy, second triple fusion, so it is called as a double fertilization. Am I clear what is double fertilization? Fertilization takes place 2 times, okay? So this is called as double fertilization. Now the fertilization done, okay? So now what are the post fertilization event, guys? What are the post fertilization? Once the fertilization over, then what are the next event takes place here? Yeah, any doubt till here? Okay.
अब एग्जाम की तैयारी को और बेहतर करने के लिए हमारे चैनल को लाइक, शेयर और सब्सक्राइब करना ना भूलें।